We have some more physiology from Dr. Hudson, and we're going to be talking about excitation, contraction, coupling today for the ECC. Now, I previously posted a video related to the neuromuscular junction, and if you haven't watched that yet, you should go back and watch that first, because it describes the first step in muscle contraction. It talks about the fact that we need to send an action potential down from the brain, from our somatic nervous system, down the axon to the terminal bouton, which is sitting on top of that muscle fiber. In that terminal bouton, we have vesicles filled with acetylcholine. The action potential will activate voltage-gated calcium channels on that bouton, allowing for calcium influx. Once calcium levels inside the bouton increase, they help drive exocytosis of acetylcholine out into the synaptic cleft, where they then diffuse across that cleft and they bind to nicotinic receptors, which are these non-specific monovalent cation channels that allow sodium influx and potassium efflux, but they allow sodium to rush into the cell faster than potassium can leave, leading to a net positive charge on the inside of that motor end plate, and we call this an end plate potential, or an EPP. And that end plate potential is going to travel up to the sarcolemma, and that's the end of the neuromuscular junction. Where does it go from here? We're going to start with excitation, contraction, coupling. Excitation as in electricity. So we need to excite this tissue again. We need to create new action potentials, and that's going to occur right here on the sarcolemma, and it's going to travel across that sarcolemma membrane, and it's going to rush down into every single T-tubule, which now penetrate down deep into that muscle fiber, and it's going to spread that electricity through the entire muscle, where it's going to have an action from there. So first and foremost, let's look right here in the sarcolemma and remind ourselves of a few things. I've got a red marker over here I'd like to use. All membranes of the human body have sodium-potassium ATP pumps. And those sodium-potassium ATP pumps are constantly pumping sodium out and potassium in. Sodium out, 3 sodium out, and 2 potassium in. Those are the sodium-potassium ATP pumps. I don't need to label them. If you made it this far into my lectures, you already know about these. They require ATP for their function, and what they do, what their physiological role is they are always creating a concentration gradient for sodium and potassium. In other words, sodium is always higher outside the cell, potassium is always higher inside the cell. And if we had a channel for these ions open, those ions can now move from an area of high concentration to low concentration. We've created a concentration gradient for these ions to run down. Okay, so out here on the sarcolemma and a few other tissues throughout the body, like the axon, what we have voltage-gated sodium channels. And we have voltage-gated potassium channels. And they are lining this. I'm just drawing two because of room limitations. They're all over these membranes. Please don't forget that. They're even down into the T-tubules. So these are voltage-gated potassium channels. So what do we know opens up voltage-gated channels? Voltage. And check it out. That's what we created with the end plate potential. Remember, potential, when we're dealing with science and physics and physiology, we need to be thinking about a voltage when we hear the word potential. So now voltage makes it up to the sarcolemma, and it will activate the voltage-gated sodium channels, opening up the gate, and now sodium can come rushing into the cell. And as sodium rushes in, that membrane starts to gain positive charges on the inside of the cell, which is when we're looking at these membrane potential graphs that hopefully you learned earlier this semester. You'll remember that we set a resting membrane potential, and that's homeostasis. It has to do with potassium leaking out of the cell faster than sodium can leak in through leak channels. But when we open up voltage-gated channels, we get a torrent. We get a rush of these ions that are crossing the membrane, this rush of current. And so if we're trucking along at resting membrane potential and all of a sudden we have a whole bunch of sodium enter this cell, what charge is sodium? Sodium with a plus charge. So plus charges are entering the cell, therefore the inside of the cell starts to become more positive. Okay? Well, that's with the EPP, and when voltage-gated sodium channels open, they're opening right here at threshold. Threshold means the opening of voltage-gated channels. So the EPP, and let me change the color so this is more clear to some of you. The EPP is going to be the first portion of this curve. It's going to be the incline of that curve. It's what allows this cell to go from resting membrane potential to threshold. But at threshold, voltage-gated sodium channels are now open, and sodium comes rushing into that cell, driving a depolarizing step, causing this cell to shoot up to about plus 30 millivolts. That's called an action potential. Okay? This is this action potential. It's the coursing of this current across that cell membrane, and positive charges now gaining on the inside of this membrane here. It moves to the next voltage-gated sodium channel, opens it, moves to the next, opens it, so forth, so on, and therefore we create an action potential, another lightning bolt that can travel down into T-tubules, and it can also travel down the sarcolemma. Okay, so this action potential is going to now travel through the entire fiber. What are those voltage-gated potassium channels all about? Well, this thing needs to inactivate somehow. Action potentials are only so long, right? They're only occurring for a very short period of time. So here at peak potential, the voltage-gated sodium channel, the inactivation gate's going to slam shut. We got a gate that's going to close. And now voltage-gated potassium channels open. They fully open at this point. So potassium can go rushing out of the cell, and we have potassium efflux, which now returns everything down to resting membrane potential. There's a lot more to this curve, this action potential curve, but I'm going to discuss that more in a video for the nervous system, not here in the muscle system. So let's go back to this action potential that's now rushing down through the T-tubules, because there are more voltage-gated sodium channels through here. And what they're going to do is they are going to start to activate another protein found throughout the T-tubule. This protein here is called dihydropyridine. And we abbreviate that DHP. So this is a DHP receptor, and it is a voltage-regulated receptor. So this protein is going to be activated by voltage. And the way it works is it's actually linked to another receptor on the inside of this cell by a chain-like protein that when it gets activated, it's going to coil that protein up. It's going to coil that protein up, and that's going to come into play in just a second. So let's draw that coil on here. Okay, let's draw this coil onto our DHP so we can think about it. All right, let's look at the rest of the anatomy of a muscle fiber. And inside of that muscle fiber, remember the functional unit of a muscle fiber is the sarcomere. But sitting on top of all of the sarcomeres, all these myofibrils inside of the muscle fiber, we have this interesting structure. I can't draw it for the life of me. So I kind of just draw a gigantic radiator drill. It just helps me because I'm not a very good artist. But this is the sarcoplasmic reticulum. This is the SR. And where the sarcoplasmic reticulum abuts the T-tubules, it's enlarged, and we call this the terminal cisterna. Okay, so the T tubules and the terminal cisterna are right next to each other. And this coiled spring-like element of the dihydropyridine is attached to another gate that's found on the SR. This gate will be opened by the coil. Okay, this is called the ryanidine receptor. Ryanidine receptor. And we abbreviate that capital R, lowercase y, capital R. This is the ryanidine receptor. And it's a calcium channel. Very important. Actually, in most cells of the body, this is a calcium channel that's activated specifically by calcium. It's called a calcium-sensitive calcium channel. But in this particular system, in the skeletal muscle system, it's now activated by DHP. So when this gate opens, if it's a calcium channel, it can allow calcium to move around. Well, what do we have right here covering the surface of the sarcoplasmic reticulum? We have lots and lots of calcium pumps. Lots of calcium pumps. And remember, a pump is a protein that uses ATP to move things from an area of low concentration to high concentration. And the sarcoplasmic reticulum is just a big bag of calcium. It's a huge bag of calcium. So there's calcium inside of here, and lots of it. Tons of calcium. Just abundant supply of calcium. Even more calcium out here in the terminal cisternas. So calcium pumps are moving things from an area of low concentration to high concentration. So calcium's always being pumped into the SR. 24/7. Every microsecond of every day, calcium's being pumped into the SR in exchange for hydrogen, if your professor cares for you to know that or not. So where is calcium concentration going to always be high? Inside of the SR. What allows it to always be high inside of the SR? The calcium pumps. So now let's go back to when the ryanidine receptor opens, and it's a calcium channel. When the ryanidine receptor opens, where can calcium go? It will rush now out of the SR into the ICF, into the sarcoplasm. And we draw it like this because it's easier to show that calcium comes down here, and therefore the intracellular concentrations of calcium, brackets means concentration, subset ICF, the intracellular concentrations of calcium are increasing. It's just easier to draw that way. But remember that the myofibril is wrapped up by this sarcoplasmic reticulum. So when the calcium is released, it's actually released onto the myofibril underneath this and all the sarcomeres that are there. So that calcium now goes down onto those sarcomeres. And that's going to begin the next step, the next video, which has to do with cross-bridge cycling. This calcium is going to go bind to a really important protein called troponin. And that allows for the contraction. So excitation, making new electricity, releasing calcium, calcium binds to troponin, and it's going to result in contraction once the myosin and the actin couple with one another. Again, that will be in the next video related to cross-bridge cycling. Hopefully that was helpful. Humankind, be both. See you in the next video.